Hello, today we are diving into transport. Now, when I say transport, I mean transport through the cell membrane. So we're just going deeper and deeper into this idea. We've already seen some of this happening in our egg lab, but we're going to continue to learn a little bit more about it so that you have some really finalized notes on it so that you can refer back to this to better your understanding. So before we begin, let's talk big ideas. So we already know that transport is materials moving in and out of the cell. This is like the whole idea of the cell membrane, right? It allows certain things in and out of the cell. And by allowing things in and out, that's this idea of transport. So movement within the cell membrane, in and out, happens because of what's called a gradient. And a gradient just means two areas having different concentrations or amounts of something. And think about it like this. If you're standing in the corner of a room and you spray a bunch of perfume, you're going to have a high concentration of perfume in that corner, but the opposite corner from you will have a low concentration of that perfume because it's just far away, nearby areas, right? Opposite corners in a room, but different concentrations or amounts of that perfume in the air. The idea here for cellular transport for the first part that we're about to talk about is that you want to get those concentrations to be equal. So let's talk about one way to equalize those concentrations. In this case, we're talking about diffusion. So diffusion is the movement of a substance. It could be sugars, it could be salt, it could be perfume, it could be food coloring. A substance from a high concentration to a lower concentration until equilibrium is reached. Okay, so this would, going back to my perfume example, this would be from the corner you sprayed in, that's your high concentration, to the opposite corner, that's your low concentration, and once that perfume moves equally throughout the entire room, you have reached equilibrium. Okay, so equilibrium is when the concentration is equal in all areas. In diffusion, substances will move from high amounts to low amounts. This is like a key term. You should absolutely highlight this, underline it, bold it, put stars next to it, whatever you want. But substances will move from high amounts to low amounts, right? High amount in this little dot. By the end, it has a low amount in that little dot, right? From high, it will move all the way to where there's none, high to low. Okay? High to low. Write that down, please. Now, osmosis is pretty much the same thing as diffusion, except it's strictly the movement of water. So everything else is the same, right? Moving from a high concentration to a low concentration through the membrane until equilibrium is reached. It will move from a high amount to a low amount, but it's the movement of water. Now, in this case and in this example, you see how there's all of these blue dots. These blue dots are the water molecules, but the sugar is not balanced, right? The sugar is not balanced in this beaker. And so for water, it's going to move until it has an equal concentration in relation to sugar. So I know there's only one sugar molecule here, so there's a little bit less water compared to the 4 sugar molecules on the right-hand side, which needs more water to hit equilibrium in relation to the sugar. I know that's a little bit confusing, but we're going to practice this in class, so don't you worry. Another type of this movement is called facilitated diffusion. And facilitated diffusion is just diffusion with a little bit of help from an outside source. In this case, it's the help of a transport protein, so one of those proteins that's found in the cell membrane. So this is used to move molecules in and out that are polar or large or charged with some sort of ion Again, molecules will move from high amounts to low amounts until equilibrium is reached. But in this case, it's just moving molecules that won't normally fit in the cell membrane. It will move it through the protein and make it a little bit, it's basically diffusion with one extra step of moving through a protein. Now, active transport. Notice how everything I've talked about so far, it just goes from high to low. It's easy peasy, no issues, right? We're just hitting equilibrium and we're good. Active transport is the opposite of that. Okay, so now these molecules are moving against the concentration gradient. This is like running uphill. It goes against the grain here. And to do this, you need energy, or ATP, right? Energy that's created in the mitochondria is used to allow this transport that goes against the gradient to happen. So in this case, you're doing the opposite. You're going from a low amount of something to a high amount of something. So in this case, you see how this is already low. There's only 2 diamonds here, but on the other side there's 5 diamonds, and this is using energy to move 2 additional diamonds out into where there's already a high amount of those diamonds. So it's kind of forcing the movement against the concentration gradient. And the only way to force something is to use energy to do that. There's a few types of active transport. The one that we just talked about with those diamonds, that's called a protein-gated channel. And so it uses those proteins that we talked about in facilitated diffusion, but it's gated, which just means that you need energy to activate it, to get it to move. So protein-gated channels, that's one. And then endocytosis, which is when large particles enter the cell, right? Enter. Endocytosis, so these like red dots are coming in and we're creating a vesicle. Or exocytosis, where large particles leave the cell, right? Exo, exit, hopefully that helps you remember. And this is when a vesicle will come up to the edge of the membrane and then like release all of whatever it's carrying. So let's just review a few things. We have passive transport. Okay, passive transport does not require energy, okay? So this is like small molecules, oxygen, carbon dioxide, water, sugars, and it moves with the gradient, right? So like you're going down a hill. You don't have to put in any energy because you can just glide down that hill, right? This is like what we've been talking about for most of the time, right? Diffusion, osmosis, even facilitated diffusion. All of this goes with the gradient, right, from high to low, and no energy. Please, please, please bold that, highlight it, underline it, with the gradient from high to low and no energy. And then active transport is the opposite of this, okay? So this does require energy, again in the form of ATP. This is like large molecules like proteins and charged particles. These are things that do not naturally just flow through the cell membrane. You need to put a little effort in to get things in and out. So this goes against the gradient from low to high. So this is like going up the hill. This is, you gotta like really, put some effort in here, okay? Examples of this are endocytosis, exocytosis, and gated channels. I know that this was a lot, and I know that this may be a little bit confusing, but don't worry. We are going to practice a ton in class so that you guys are going to be absolute pros at this. All right, good job. I will see you in class.